All right, let's talk about something incredible. A silent, microscopic engine that's running your entire life, right now. It's called the cell cycle. This is the process that literally builds us from a single cell, heals our wounds, and just keeps our bodies running with a kind of precision that is absolutely mind-boggling.
I mean, just think about it. In the few seconds it's taken me to say this, your body has already produced millions of new cells. So how does it do that? Well, the answer is this ancient, incredibly organized mission that every single one of your cells is programmed to carry out. So let's dive in and follow that mission right from the very beginning. Okay, so before we get into the nitty-gritty of how this all works, we really need to understand the why. What is a cell's grand purpose in the first place, and why is splitting in two such a critical part of its job description?
When you really boil it down, the cell cycle is a journey with a very specific, crucial destination: to create two perfect, identical daughter cells from one original cell. You can think of it as the ultimate act of cellular inheritance, right? It's all about passing on that precious blueprint of life.
And believe me, this journey has a huge purpose. For complex organisms like you and me, it's the engine that drives our growth, and it's our built-in repair system. But for single-celled organisms, this cycle is reproduction. It's how they create a whole new generation. So to put it simply, this process is absolutely essential for pretty much all life as we know it.
Okay, but a cell can't just split on a whim. Before the main event, the actual division, it has to prepare. And this is where we get the cell's training montage. It's a phase called interphase, and it's where all the critical prep work gets done. In fact, this is where the cell spends most of its life, just getting ready. And this isn't a quick thing. Interphase takes up over 90% of the entire cell cycle, and it's split into three main stages. First up is the G1 phase. This is where the cell just grows. It bulks up, makes more proteins, more organelles. Then comes maybe the most critical step of all, the S phase, which stands for synthesis. This is where the cell painstakingly copies its entire DNA blueprint. And finally, you have the G2 phase, where the cell does one last final growth spurt, double-checking that everything is absolutely perfect for the big split.
Okay, let's just pause on that S phase for a second, because the scale of what's happening here is, well, it's staggering. We're talking about a cell duplicating a genetic code that is over 3 billion letters long, and it does this with near-perfect accuracy. Because a single mistake, that can have really serious consequences. It is a monumental feat of biological engineering, and it's happening in trillions of your cells right now.
So after all that intense preparation, you'd think, okay, the cell's ready to go, right? Time to divide. But not so fast. Before it gets the green light for division, it has to pass a series of really rigorous, super high-stakes inspections.
See, the cell cycle has these built-in safety mechanisms called checkpoints, and they're exactly what they sound like. They are points where the whole cycle just pauses to check if everything is correct before it's allowed to proceed. It's basically the cell's internal quality control department making absolutely sure it doesn't pass on any damaged goods to the next generation. And this brings us to a real moment of truth, the G2 checkpoint, which happens right before division is supposed to begin. The cell essentially asks itself a critical question: is my DNA replication complete and error-free? If the answer is yes, fantastic. It gets the green light to proceed to mitosis. But if the answer is no, it is a hard stop. The entire cycle halts, repair mechanisms kick in, and honestly, the cell's fate really just hangs in the balance.
And this rule is absolutely non-negotiable. You can think of the G2 checkpoint as a super strict gatekeeper. It simply will not allow a cell with faulty DNA to divide, period. This is what prevents the spread of harmful mutations, and it's one of the most fundamental safeguards we have for our genetic integrity.
All right, so once our cell passes that final critical inspection, the main event can finally begin. And this is mitosis. It's a beautifully choreographed, incredibly dramatic process where all those duplicated chromosomes get sorted and separated. It really is like watching an intricate dance.
So, Act 1 of this dance is called prophase. Here, all that long, stringy, duplicated DNA starts to condense and coil up super tightly, forming those classic X-shaped chromosomes you see in textbooks. At the same time, a structure called the mitotic spindle begins to form. And a good way to think of this is like a set of molecular ropes that are getting ready to pull the chromosomes into place.
Next up is metaphase, and this is just a stunning moment of perfect order. Those spindle fibers, those molecular ropes, they attach to each and every chromosome, and they guide them to line up perfectly. A perfect single-file line right down the equator of the cell. This alignment is so crucial because it's what guarantees that when the chromosomes finally split, each new cell will get exactly one copy of every single one.
And then comes the most dramatic move of the entire dance, anaphase. The proteins that were acting like glue, holding the two identical halves of each chromosome together, those are called sister chromatids, they just dissolve. Instantly, the spindle fibers contract, pulling the chromatids apart in this powerful, decisive motion towards opposite ends of the cell. The dance partners have officially separated. The grand finale of our dance is telophase. As the two complete sets of chromosomes arrive at their opposite destinations, new nuclear membranes start to form around each group, like a protective bubble. The chromosomes themselves start to unwind and decondense, relaxing back into their normal stringy state. And with that, mitosis, the division of the nucleus, is officially complete. What we have now is one cell, with two perfectly identical nuclei inside. So we've just walked through this perfect, incredibly orderly process. But what happens when this intricate system breaks down? What happens when the cell just loses control?
You know, this is probably one of the most important questions in all of modern biology. Because when a cell just plows right through a checkpoint, totally ignoring the stop signals and the warnings about DNA damage, the consequences, well, they can be absolutely catastrophic.
And the contrast here is just stark. A normal healthy cell is highly regulated. It stops when it detects errors and it fixes them. A cancer cell, on the other hand, has completely lost that control. It divides relentlessly, it blows right past all those safety checkpoints, and it passes on its genetic errors to every single new cell it creates. And this completely unchecked division is exactly what leads to the formation of a tumor.
So, you see, the cell cycle is truly the foundation of our existence. Understanding this intricate dance is the key to life itself, but it leaves us with this huge, ultimate question. Can we ever become its master? Can we learn to control it, to fix it when it breaks? The answer to that question, well, that's going to define the entire future of medicine.